presentation is study of total phenol, protein, lipid, and carbohydrate content in selected varieties of the potato, which is grown in the Northeast region. So particularly, I have taken 4 districts, I have chosen 4 districts from Manipur, and I have done this particular experiment. So my objective is to find out the total phenolic, protein content, lipid, and carbohydrate content present in the local variety of potato. So this local variety is known as Abertabi, which is red in color. Its size varies from small to a little medium size, but we cannot find a very good large variety in this potato. And second objective is after finding out the different percentage of this nutrient value, we can encourage the farmers to grow more of these varieties for the benefit of the people. So my introduction part is this particular type of local variety is one of the richest source of calories needed to maintain day-to-day output of the human energy. It is a valuable food for those who seek to lower their blood pressure. Particularly, I have mentioned here, it is a valuable food for those who seek to lower their blood pressure. And this protein has a very relatively high nutritional quality. They have a quick digestibility and low residue of cooked potato, makes it a valuable food for infants. The potato is widely cultivated both in the hills and valley areas of Manipur, and the taste differs due to the climatic condition and it may also be due to the soil variation, because many studies have reported the different nutrient content in the soil, and so due to the difference in the nutrient value, maybe the percentage of the protein level or lipid level or the carbohydrate level vary. So collection was done during the summer season of 2020 and 21. And the district that I had selected for the study were Kakching district, Imphal West district, Kangpokpi district, and Senapati district. These samples were collected for analyzing the total protein, carbohydrate, phenolic, as well as the total lipid content. So the sample preparation is, it was uniformly, it was first washed out thoroughly with the tap water, and then the peels were removed and they were sliced into about 4 to 5 millimeter slices or cubes, approximately 1 centimeter size. The sliced tubers were set dried, because you cannot dry it directly in the sun. It has to be set dried. The dried pieces were grounded to coarse powder or into a fine powder with the aid of a blender, and it is used for the chemical analysis. So there are different assays we have to follow. There are different protocols. So for this phenolic content, I follow this CO method. So here, the alicot of the extractor standard solution of gallic acid, it was taken in 1, 2, 4, 5, 6, 7, 6, 8, and then 10 microgram per liter in a test tube and made up to the volume of 1 ml with distilled water. Then about 0.5 ml of the following CO culture reagent, which is in the proportion of 1:1 with distilled water, and 2.5 ml of 20% sodium carbonate solution were added subsequently to the test tube. The reagent blank was prepared because we have to compare it with the blank in order to find out the actual value of our particular reaction. So it was used with distilled water, and the reaction mixture was mixed well, and then it was kept for incubation in the dark at the room temperature for about 1 hour, and the absorbance against the prepared reagent blank was determined at around 725 nanometer with the UV visible Shimazu spectrophotometers. And so for this particular reaction, we had used the Shimazu spectrophotometer. All the determinations were performed in triplicate so that we can get a correct value. So for the estimation of protein, we have chosen Gelda's method, which is a very important method, and everyone has used this method because it is a standard one. And for this method, it has 3 steps. The first is digestion, the second is distillation, and the third one is titration. Let me elaborate a small procedure about the digestion. So the tubes were loaded with about 0.3 gram of plant sample, 10 ml of concentrated sulfuric acid, and 3 gram of catalyst mixture. It was a mixture of potassium sulfate and copper sulfate, which is in the ratio of 5:1 in an insert rack. Then place the manifold over the tubes and loaded the insert rack and the manifold in the digestion block. The temperature was set initially at about 250 degrees Celsius, and if protein was not there, it was increased to about a temperature of 50 degrees more in the gap of about 15 minutes and up to about 400 degrees Celsius. The digestion time was approximately 2 to 3 hours, and at the end of the digestion, we know that when we see some condensation reaction is going on, we know that the reaction has ended, and at this point of time, we can get ready for distillation process. Again, the procedure for distillation is first, the conical flask was loaded with about 20 ml of 4% boric acid and 3 drops of mixed indicator in the receiver side, which will be in the pink coloration initially. Then the digested sample was loaded and then added about 40 ml of the 40% sodium hydroxide solution. The time taken for the process was about 9 minutes, and we know that after 9 minutes, the color in the conical flux changes from the pink color to the green color, and this is the end point of the distillation of the sample. So when this green color color of this sample changes into green color from pink, we know that the reaction has ended, and this is the end point of the distillation process. So next is the titration process. The distillated solution was titrated with using 0.1 normal high CL solution till the color changes from green color to the permanent pink color. So in this step, the color which was changed from pink to green is finally changed again into the permanent pink color, and this indicates that the whole titration has ended, and this is the end point of titration. So we have to estimate the total carbohydrate content using this Entrone method. So this is the method. This Entrone reagent was freshly prepared before used by dissolving about 200 milligram Entrone in 100 ml of the ice cold sulfuric acid, which is about 95%. The stock solution of standard glucose was prepared by dissolving 100 mg in the 100 ml of water. Working standard was made by taking about 10 ml of the stock and diluted it to 100 ml with distilled water. It was stored again in the refrigerator and after adding a few drops of toluene. So for the lipid estimation, I have used this Blight and Dyer method, and here we have done the experiment like this. 5 grams of the potato was homogenized in 10 ml of methanol and added about 5 ml of the chloroform. It was again homogenized for again another 2 minutes. Then 5 ml of the additional chloroform being added and homogenized again for about 30 minutes. The 5 ml water was added and homogenized for another 30 seconds again and filtered through a Buckner funnel with slight suction, and the liquid was retained. These liquids were combined and transferred into a separate funnel and passed the chloroform layer through a 2.5 centimeter thick layer of anhydrous sodium sulfate using Whatman number 1 filter paper, which is a little bit different from the usual filter paper. And then it was washed with about 20 ml, which is about 1:1 ratio of chloroform and methanol in volume by volume. So the solvent was removed using a rotatory evaporator under vacuum at about 40 degrees Celsius, and the calculation was done using the following formula. So now the result obtained was, here I have represented, I have given the protein content, carbohydrate content, total phenolic content, and the lipid content in these 4 columns. And these are Senapati, Kakching, Kangpokpi, Imphal West. These are the districts of selected districts of Manipur, where I have collected those potato sample, that particular local variety of the potato, which is also called as a bear tribe, which is pinkish in color. So from this table, we can understand that the maximum amount of protein is present in those potatoes which are collected from the Kangpokpi district. And then for carbohydrate, again, this minimum amount of carbohydrate is present in those sample of potato which is collected from the Senapati district. And finally, for this phenolic content, it is almost same. And when we compare the study of the phenolic content with the other types of other varieties of potatoes which have been reported by different researchers, it comes around these ranges. That means there is not much fluctuation in the phenolic content of the potato varieties, whether it be our local variety or the other type of white variety, or those varieties which are grown in other different parts of the country. They almost report the around about 200 something of this phenolic content, and lipid content also, it does not vary. But what we see here in my study and the other study is that usually the protein content of the varieties of potatoes which have been studied by the researchers, they report very less amount of the protein content. But in this particular variety, Aberchavi, which is the local variety of our local variety of this potato, we see that the content of the protein is above 8%. But in other potato variety, we come across about, it will be around 7 or sometimes 4 to 6 like that. So this is the main reason. This may be the main reason why I am showing it in the next slide. Okay. So here I have, in this slide, I have given this uses of the red variety. So maybe due to the high percentage of the protein content in that particular variety, postnatal women, that is those women who have given birth or after delivery, they are usually in Manipur, they are given this particular red variety of potato in their diet as a supplement for protein content, because I don't know the reason why people, they used to have this protein, particular variety of potato for this postnatal women. But after my study, I came to know that maybe because of the high protein content, those people, they don't have any idea about the protein content. But after study, I came to conclude that maybe because of the high percentage of protein, they have given this particular variety of potato to the women or to the postnatal women. And even for a diabetic patient, they have chosen this particular variety, maybe because the carbohydrate content of this particular variety is very less compared to the other varieties which are found in different parts of the world. These are as per the research papers that I have done, that I have I have been checking as a reference. So and again, I can also give another conclusion that when we check the protein percentage of protein of the four different districts of Manipur, we have I have undergone through some literature survey about the soil nutrient content of the variety of the soil varieties which are of the particular regions which I have selected. So among them, I have noticed that this Kangpokpi area, where the protein content is very high, it has got maximum nitrogen content compared to the other three parts of the other districts of Manipur. So maybe the nitrogen content is very high in this soil, so maybe the protein content is very high. So this is what I have concluded. And so this observation is difference in the protein content of the sample cultivar in different districts, maybe due to the difference in the nutrient content in the soil and of the different districts under study. These are the references, and thank you all for your patience hearing.
Thank you for your wonderful presentation.